You have Duchenne's, and you have your Becker's muscular dystrophy. All right, so this is how this is our this one, the structural proteins. Dystrophy with the dystrophin complex is very important to understand. Now we have the other thing, which is a concept called triads, and we'll compare it with something called dyads versus dyads. And what are your triads? So let's focus over here. Your skeletal muscle, your sarcomeres, have something called triads. So you know how your skeletal muscle over here has a connection with the nerve. So here's your sarcomere. Okay, here's your sarcomere, your muscle fiber cell, and you have a nerve over here, alpha motor neuron, which releases acetylcholine, and those will cause an action potential in the sarcomere. Well, this action potential has to go into the cell, deep into the cell. So for that, and once the action potential goes deep into the cell, calcium has to increase in the cytoplasm. So how is that supposed to happen? We should discuss that now. So we have these things called triads. A triad is made of two types of tubules. So this is your sarcomere, which you could see in this picture over here, and so let's just remove this. It's okay, it's fine. So you can see the thick filaments over here. This is the myosin coming from the M line over here, and these are the Z lines over here, you could see over here. So these are the ends of the sarcomere. I'll just extend this over here a bit. Now look at the AI junction, at the junction where the A band and the I band meet. The sarcomere takes a dip over here. There's a sort of well-like structure which is in red over here. So this is your sarcomere. This is red in color, and it goes deep down over here. This is your sarcomere. This is the the cell membrane, sarcolemma, and in this there is a sort of well, and this well is called the T tubule. What do we call this? A T tubule. So it's like a tubule which is like a well, which, so the action potential that's formed over here on the cell membrane is going to go down this T tubule to the very end of this T tubule. Okay, so these are your T tubules, and then in green over here we've got these things called L tubules. What is an L tubule? This is your smooth endoplasmic reticulum, or your sarcoplasmic reticulum of the muscle, and it extends longitudinally along the length of the muscle cell, along the muscle fiber. Can you see it's long? It's not transverse. T is for transverse, L is for longitudinal. Tudinal. T is known as transverse. So you have your T tubule and you have your L tubule over here. So you see these L tubules over here, and you have your T tubules over here. Then what happens is these L tubules are your smooth endoplasmic reticulum, and the T tubules are just like wells, like an invagination of the cell membrane that goes into the cell. So let's magnify this image over here, and we'll have a look at what's going on over here in this part. So this is your T tubule, right? This is your T tubule, and the action potential goes down, down, down after you make the action potential on the neuromuscular junction area. And now down the T tubule you have this protein. This is a sensor, a voltage sensor, and this is called dihydropyridine. Okay, in the cardiac muscle it's a voltage gated calcium channel, but here in your skeletal muscle it's just I want you to think of it as a sensor. Okay, so it's a sensor in your skeletal muscle which senses the action potential, the voltage gated sensor, the voltage sensor, and as the action potential goes down, the inside of the cell is positive. And you should watch the action potential video to understand this. Please watch that video, it's important, and it's very beautifully explained, and I hope you all watch those other videos. You can't understand neuromuscular without knowing the general physio. So we have dihydropyridine receptor, and these are called dihydropyridine because they're blocked by a substance called dihydropyridine. So if you are aware, there is something called calcium channel blocker. This is a class of drugs that we use to lower the blood pressure in patients, and sometimes to even slow down the heart. And how there's a class of calcium channel blockers called dihydropyridine blockers. So they block the dihydropyridine. Okay, so anyway, dihydropyridine blocks this, so that's why it's called dihydropyridine. Anyway, the voltage that changes because of the action potential activates this dihydropyridine, which is at the end of these T tubules. So you have them on both sides. And now what happens is, next to a T tubule there are two L tubules over here at this AI junction. When the cell is in resting state, you can see. So you have two L tubules over here, one on each side. This is an L tubule, this is an L tubule, and this is a T tubule. So this is how you got the triad, three tubes coming together. So this is the T tubule, and it has dihydropyridine on it. And now you see the L tubules next to it. This is an L tubule, this is an L tubule. So you have L tubule over here, you have L tubule over here. Alright, now these L tubules are the smooth endoplasmic reticulum, and there's a calcium channel which is called ryanodine receptor. Ryanodine, ryanodine. Okay, so please remember this, ryanodine receptor. And what happens is dihydropyridine receptor is attached to ryanodine with something called the foot process in your skeletal muscle. In your skeletal muscle, not in your cardiac muscle. Cardiac muscle is different, okay? Cardiac muscle has dyads, they are different. I'll talk to you about that later. The point is, this dihydropyridine has a connection with the ryanodine receptor. Now, when an action potential comes down and makes the area around the dihydropyridine positive, meaning it depolarizes, it will cause this foot process to move, boing, and it'll just pull the foot process back. So think of it like what happens is, there is this dam. Okay, so there's a dam, or there's a big water tank, and there's a lot of water in this tank, and there's a hole in this tank, and so water tries to come out. So what happens is, if you try to block it with your hand, ryanodine is your hand, and this L tubule is the smooth endoplasmic reticulum with a lot of calcium stored over here. So there's a lot of calcium in your smooth endoplasmic reticulum. Well, what happens is, when you give this action potential down the T tubule, the dihydropyridine is going to pull this foot process out of the way. So it's like you pulled your hand back, and now what happens, calcium can gush out into the cytoplasm, and that can stick to the troponin C, causing the contraction of the muscle to take place. Calcium is heavily concentrated in these L tubules. And how is that done? Because there is a protein called SERCA ATPase. SERCA ATPase is present on the L tubules, and this is called smooth endoplasmic reticulum calcium ATPase pump. So calcium enters into this cell, and it will concentrate heavily. And how does this happen? The calcium binds to these proteins called calcium binding proteins, like calsequestrin, sarcolumin, histidine rich calcium binding protein. So all these different proteins are going to hold on to calcium and help in concentrating the calcium here. And when an action potential comes down, you will be able to release this calcium, because there's so much concentrated because of the SERCA ATPase. That means when the action potential is over, SERCA ATPase pump will pump the calcium back from the cytoplasm into the L tubules, causing relaxation of the muscle. So relaxing a muscle is an active process. You are keeping that calcium in at the cost of ATP energy. So you're using one of those primary active transfers, those pumps over there, to pump calcium inside. Anyway, so when you give an action potential, you release this calcium. This is the triad which you have in your skeletal muscle. In your cardiac muscle, it's a little different. I'll just talk to you about that over here in this little space. Cardiac muscles, they're different from skeletal muscles. So look at this. You have two AI junctions in each of your sarcomeres. So you have two triads in each skeletal muscle. You have one triad here, one triad here at the AI junction, at the A band and the I band junction over here. So in the cardiac muscle, let's say this is a cardiac muscle. You know that one sarcomere is like one cardiac cell. They don't fuse. So cardiac muscles are quite small, and they're uninucleated. They're single nucleated. And here we have the T tubule, which enters at the Z line, and one L tubule over here along the entire length of the muscle. This is your dyad. You have a T tubule and you have an L tubule. And there's a little, let's look at this picture a little more in detail. So this is your T tubule. You have dihydropyridine over here, okay, dihydropyridine over here, dihydropyridine, and there is ryanodine over here. This is ryanodine receptor. Now there's no foot process connecting them together. What happens is when an action potential goes down the T tubule, like this, when the action potential goes down to the dihydropyridine, this dihydropyridine is a calcium channel. Dihydropyridine over here is a calcium channel. It's a voltage gated calcium channel, okay. So the calcium, a small amount of calcium is going to get sprayed on your ryanodine. What's going to happen? Calcium gets sprayed on the ryanodine receptor, and this is going to activate the ryanodine receptors because they are sensitive to calcium, and it's going to open the ryanodine channels, allowing more calcium to be released. So this is called calcium induced calcium release. Calcium induced calcium release, because some calcium comes in and then you release a lot more calcium. All right, so this is your calcium induced calcium release that takes place over here. And this is seen in cardiac muscle, and if you look, you only have one dyad for each sarcomere. So this is most of the skeletal muscle structure that we need to know, and a few differences between the skeletal versus cardiac muscle structure. And now we'll go into how these the excitation contraction happens properly. So until we watch that video, I say goodbye, and I'll see you later. Das Wadania. Bye bye.